Society treats heartbreak as a poetic metaphor or a transient bout of sadness. Neurobiology tells a completely different story. Clinically, the termination of a strong social bond registers as a physiological trauma. Acute social loss behaves as a literal injury to the nervous system, immediately followed by a severe neurochemical withdrawal state. This 3D model compares functional magnetic resonance imaging, or fMRI, data from two different states. fMRI is a technique that maps brain activity by measuring localized changes in blood flow. On the left, we see the neural response to acute thermal pain, a physical burn. On the right, we see the response to acute social rejection. The identical neural coordinates illuminate in both scenarios. The active structures here are the anterior cingulate cortex and the insula. The anterior cingulate cortex, or ACC, is a region deep inside the brain associated with error detection and affective distress. The insula is a cortical structure responsible for processing bodily perception and pain. Social loss natively hijacks the exact same somatosensory hardware the human body uses to detect physical tissue damage. Because the brain interprets a severed relationship as physical damage, it does not prompt a state of passive reflection. It triggers an aggressive, motivated physiological drive state. To map the architecture of this condition, we have to isolate three distinct neural networks working in tandem. The first network is the somatosensory pain overlap in the ACC and insula, creating the acute sensation of distress. The second network is the mesolimbic craving state, located deep in the midbrain. The third network comprises the prefrontal amygdala regulatory circuits. The amygdala is a small, almond-shaped cluster of nuclei responsible for processing fear and separation distress. Treating this condition requires addressing a multi-system biological failure involving pain, addiction, and cognitive regulation. Understanding the ACC's role in social pain requires examining a historical error. Since James Pepys began mapping the cingulate gyrus in the 1930s, neuroscientists divided the ACC into strict, mutually exclusive functional zones. The prevailing model dictated that the dorsal ACC, or DACC, located in the upper region of the cortex, handled cognitive conflict and error detection exclusively. The ventral ACC, or VACC, located in the lower region, was assigned to emotional processing. That model fractured under the weight of modern neuroimaging research conducted at UCLA. Researcher Naomi Eisenberger published a comprehensive meta-analysis evaluating 46 separate social pain studies containing fMRI data from 940 healthy subjects. Look closely at this breakdown. Experiences of rejection, whether from a digital exclusion game called Cyberball or from subjects actively reliving a severe personal loss, activate multiple ACC subregions simultaneously. These include the DACC, the pregenual ACC, or PGACC, and the subgenual ACC, or SGACC. Skeptics argued that DACC activation during a digital exclusion game was simply an artifact of expectancy violation. Expectancy violation is the cognitive surprise a subject registers when a predicted outcome fails to occur. Eisenberger's data isolates this variable. When researchers test subjects in strictly controlled conditions that eliminate cognitive surprise entirely, the DACC maintains a high-amplitude firing pattern in response to social rejection. Furthermore, activity in the pregenual and subgenual ACC reliably correlates with the severity of self-reported social distress across the entire 940 subject pool. The dorsal ACC natively processes the affective distress of social pain. The brain makes no structural distinction between the physical damage of a broken bone and the psychological distress of a severed social bond. That explains the pain. It does not explain the intrusive behaviors, the inability to stop checking a former partner's social media, or the obsessive rehashing of old memories. Anthropologist Helen Fisher designed an fMRI study to analyze individuals who were recently rejected, but reported that they were still intensely in love. Fisher's experimental design isolated the heartbreak variable by placing subjects in an fMRI scanner and showing them an alternating sequence. They viewed a photograph of their rejecter, performed a distraction task, and then viewed a photograph of a familiar, neutral individual. Viewing the rejecter bypasses higher reasoning entirely and directly activates the midbrain. Specifically, it heavily activates the ventral tegmental area, or VTA, pushing dopaminergic pathways directly into the ventral striatum. The ventral tegmental area is a group of neurons at the base of the midbrain that heavily regulates motivation and reward. The VTA and its connected mesolimbic circuitry operate as the mammalian brain system for meat choice. It processes survival-level gains and losses. This exact anatomical circuit is responsible for driving cocaine craving in addicts. Heartbreak operates as a literal dopamine withdrawal state. This sudden dopamine starvation forces the brain into a fight or flight response. It drives black and white cognitive distortions, where the subject views the relationship through aggressive extremes, completely bypassing nuanced logical assessment. Logical advice from friends fails because the addict's brain is singularly focused on obtaining its withheld dopamine fix. The brain treats the lost partner as a withheld survival reward. The heartbroken individual behaves with the exact logic and physiological urgency of a drug addict coming clean. If the brain is trapped in a loop of somatic pain and dopamine withdrawal, what mechanism allows a person to eventually recover? Recovery relies on active, top-down prefrontal control to dampen the amygdala's separation distress signals. Researchers Peter Fried and his colleagues at Columbia University mapped this regulatory circuit by studying subjects experiencing acute pet bereavement. They used an emotional Stroop task to measure subconscious attentional bias. The subjects were shown rapidly flashing neutral control words, contrasted against words related to the deceased, and the researchers tracked the microfluctuations in reaction time. The resulting reaction time bias proves a structural reality. Grieving subjects cannot physically stop their attention from being hijacked by reminders of their loss. The fMRI data revealed a double dissociation discovery. Two distinct psychological coping styles map perfectly to two opposing neural circuits. Subjects reporting high levels of intrusive thoughts exhibited a specific neural signature, hyperactivation in the ventral amygdala and the rostral ACC, or RACC. The RACC is a specific frontal subsection of the cingulate cortex, highly involved in emotional processing. Conversely, subjects who exhibited avoidance behaviors showed deactivation in the dorsal amygdala and the dorsolateral prefrontal cortex, or DLPFC. The DLPFC is an area of the brain responsible for executive functions and high-level cognitive control. The study isolated the recovery mechanism. High functional connectivity between the DLPFC and the amygdala successfully dampens attentional bias, allowing the subject to regulate their distress. Clinical complicated grief is not a failure of willpower. It is a measurable failure of functional connectivity between the executive prefrontal regions and the primitive amygdala. By synthesizing the data from Eisenberger, Fisher, and Fried, we can map the complete architectural model of the heartbreak state. At onset, the VTA initiates an aggressive craving state via a severe dopamine drop, while the ACC registers the severed social bond as a critical somatic injury. This dual system alarm triggers an immediate release of cortisol. Cortisol is the body's primary stress hormone, and its sudden influx drives the physiological symptoms of shock, including sleeplessness, elevated heart rate, and immune suppression. The resolution phase relies entirely on active, computationally expensive prefrontal regulation. Successful regulation occurs when the prefrontal circuits contextualize the event, projecting inhibitory signals downward to suppress and dim the lower brain's distress signals. Surviving this state requires the highest-order cognitive networks to physically overpower the brain's most primitive survival mechanisms. Why would the human brain evolve a mechanism that makes the loss of a social bond so physically and cognitively agonizing? For a prehistoric mammal, isolation meant certain death from starvation or predation. Survival was entirely dependent on the cohesion of the group. Social pain is a highly conserved evolutionary alarm system, engineered to prevent that isolation. The physical pain and the dopamine withdrawal act as biological imperatives. They force the organism to seek reconnection and return to the safety of the group. When an individual experiences the total systemic overload of relationship loss, they assume their brain is breaking down. In reality, the brain is functioning exactly as designed. Heartbreak feels like dying because, to our primitive neuroanatomy, isolation is death. The agony of rejection is the exact mechanism that kept our ancestors alive.